Hi Ali, am I audible?
Yes, sir.
Okay, so in the last class, we talked about this globular protein and fibrous protein. Do you have any doubt in the previous topics that we have discussed so far?
No, sir.
Okay, so let us move ahead and uh, can you please open your web camera so we can start? Thank you. So now, in the last class, what we did, we classified proteins on the basis of their molecular structure, right? Now we are going to talk about the structure of protein itself. So we have four categories. We will go into them one by one. The first one is primary structure of protein. So let us talk about the definition first, then I will show you the figure. It refers to the sequence in which amino acids are arranged in protein. Each protein has a specific sequence of amino acids which is critical for its biological activity. Any change in the sequence of amino acids create a different protein itself. Right. So what we are doing here is we have 20 types of amino acids. We are making a chain of it. If I am changing the sequence, I am getting an entirely new protein with a different type of biological activity, right? So basically what we are talking is, see, this is what? These are amino acids and they are arranged in a specific sequence. And this is what? This is our primary structure of protein, like a chain type of thing, right?
Yes,
If
sir.
I am if I am replacing any amino acid with other, then I am going to create an entirely new protein with different biological activity, right? So this is what about primary structure of protein. I have wrote in Urdu as well. 20 टाइप के अमीनो एसिड होते हैं जिनसे बहुत तरह के प्रोटीन बना सकते हैं, राइट? You can make any type of protein by changing just the sequence of it, just sequence of amino acid, right? Now, what do
Yes,
we
sir.
mean by secondary structure of protein? It refers to the arrangement of polypeptide chains. So I do you know what are polypeptide chains? I told you in the last class. What are polypeptide chains? So let us talk about a simpler question. In amino acid, which type of linkage we observe? If I am connecting two amino acids, which type of linkage I am observing there?
Peptide
Peptide. If I am
linkage. Peptide.
okay, good, good. If I am talking about specifically two amino acids, uh, which type of peptide linkage is that? For if I am connecting two amino acids, the linkage is peptide linkage. I know, but which type of peptide linkage? Bidentate, didentate, tridentate, tetradentate. Which type
Identity.
of? I
Identity.
okay. Now, if I am talking about polypeptide chains, I mean we have at least 10 amino acids which are connected with each other. At least 10, right? So what we are doing here is it refers to the arrangement of polypeptide chains giving rise to a particular shape arises a result which arises due to the result of hydrogen bonding. We have two types of secondary structure. First one is alpha helix and second one is beta plated sheets. So we should go into the figure. It will help us to understand better. Let me correct something which arises as a result of hydrogen bonding. See, suppose you have your primary structure, right? If you are bending this state chain or bending this chain in a specific arrangement, then you are going to get secondary structure. If you are bending it even more, if you are making it more complicated, then what is going to happen? It will become into tertiary. If you are making it even more complicated, you are making a spherical type of coil, then it will become into quaternary, right? So let us specifically talk about secondary structure. See, in secondary structure, suppose this is our what primary structure, right? If I have my primary structure and I am doing what? I am making a coil type of thing, right? This type of thing, alpha helix. So this is what? This is our secondary structure. And if I'm making the primary chain into this specific kind of thing, this alpha type of form, then I will call it alpha helix. If I'm making it like this, if I'm making it like this, we will call it beta sheet. Is this clear? The shape,
Sir.
how we are going to do it. You will have a primary chain. You are just playing with it. You are making it like if alpha helix kind of thing, then you will call it alpha helix. And it is a category of secondary structure. Right. So let us talk specifically about what is happening while we talk about alpha helix structure. See, in this type of structure, the peptide chains are coiled up to form a helix. You know what is helix? A spiral kind of thing, right? So
Yes,
they
sir.
are coiling to form a helix. This helical pattern is found to be right-handed and is called alpha helix. This is helix. Basically, it is helix, and because it has a right-handed kind of rotation, then we will call it alpha helix, right? The intermolecular hydrogen bonding are formed between carbonyl group of one amino acid residue, hydrogen bonded to NH prop and adjacent. See, in these alpha helical chains, we have NH somewhere here and we have carbonyl here. Due to these two entities, they are forming which type of bonding? Hydrogen bonding. And this is also the reason why they look like this. Is this making sense? Inside this chain, we have NH as well as carbonyl group. And the hydrogen of that nitrogen is getting bonded with our oxygen and making a hydrogen bonding, right? And this hydrogen bonding is the reason why they are going into the secondary structure at the first place, right?
Sir.
Is this clear? So what we will say, the intermolecular hydrogen bonding are formed between carbonyl group of one amino acid. We have several amino acids because we are talking about polypeptide chain. Uh, Group of one amino acid residue hydrogen bonded to a NH group of an adjacent turn of the helix. Is this clear?
Yes,
Is the
sir.
language simple or do I have to make it even more simpler?
No, sir. That is simple.
This is how our alpha helix look like, and this is how our beta look like. So let us talk about beta first. I have taken different type of representation so you can understand it, right? We can draw beta like this as well and alpha like this as well. Again, these are a secondary structure, alpha helix and beta, just a different way of representation, right? So let us talk about what are beta plated sheets, right? So these look like this. We have one sheet and we have another sheet and they have bonding with each other like this. So what is happening? Let us understand. The neighboring peptide chains are held together by intermolecular hydrogen bonding, resulting in the forming of flat sheet type structure. These sheets are stacked one above the other like folded pages of the books to form 3D structure called beta plated sheets. So what is happening? You have one polypeptide chain in this manner. You have one polypeptide chain arranged in this manner, like this. And in the similar way, you have several polypeptide chains that are coiled upon each other, like they are very close to each other, and they are having hydrogen bonding. All right. And due to which they
Sir.
are forming a flat sheet type of structure. You can imagine it like the folded pages inside your book, right? You have pages, they are like, how can I, like, let me take a book first of all. Like, do you have a book? If you are folding a page and you are folding several pages, let me take a book. Just give me a second. See, Yeah, I don't know is it going to be visible or not. You have your book, you have folded one page, and just beside it you have folded another pages, right? Uh, give me a second. Try to see it now. Are you seeing these pages like
Yes,
this?
sir.
You folded
Yes,
one
sir.
page and you folded just in the same manner, just beside it. So this is how our beta type of like beta protein look like. You will fold several pages. These one page can be considered as a polypeptide chain. You have folded, you have so many peptide chains and which are attached to each other in a way they will appear as folded pages inside a book, right?
Sir.
And you can draw this structure to get full marks, right? Draw this structure as well. Now, how will you form a tertiary structure of protein? If you are making it even more complicated, if you are folding it even more, then it will convert into tertiary. How? Let us see. We had our primary, simplest. We folded it. We like made it a bit different shape, right? So we form secondary structure. These two are secondary. If I folded it even more, if I made it even more complicated, then it will be called tertiary. So let us talk about what are tertiary. See, here you can differentiate these two things. This is a bit more complicated than this, right?
Yes, sir.
More folded. You can see it over here as well. These are again regional of structure, structural helix, right? Tertiary structure, 3D overall fold of the protein containing secondary structure, region of secondary structure alpha helix, region of secondary structure beta helix. So tertiary structures have both component, slight of both component. They have our alpha helix kind of structures as well. Like somewhere they will possess this alpha helix inside their structure, and somewhere they will also possess what? Beta. So they have both. That's why they are tertiary. Is it making sense?
Yes, sir.
So let us talk about the definition. Tertiary structure of protein represent overall folding of the polypeptide chain. That means further folding of the secondary structure. Just what I said. If you are making the secondary structure even more complicated, then you will reach to the point where they are no longer secondary structure. You will call them tertiary structure. It gives rise to two major shapes, fibrous and globular. What we understand here, this fibrous and globular proteins are part of which type of protein? Tertiary protein, right?
Yes, sir.
The main forces which stabilizes the 2-degree and 3-degree structure of protein are hydrogen bonding, disulfide linkage, and van der Waals forces and electrostatic forces. Basically, hydrogen bonding is due to the carbonyl group and nitrogen group. These disulfide linkage is due to the sulfur group, which is present also, right? And van der Waals forces, the weak ones, and electrostatic forces. These are the reasons because these are the reasons uh why they are stable because of these kind of forces, right?
Yes,
I will
sir.
show you a detailed structure of these type of proteins, like how the atoms are there, which type of atoms are there, just in a while. Okay, so what is quaternary? If you are making even like more mess of that protein, the simple chain, you are making it more complicated, even more complicated than tertiary, then you will reach to the point where they are quaternary. So two or more polypeptide chains refer to as a subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. So let us see the figure. Suppose you had one tertiary polypeptide chain. You have this polypeptide chain. You bout another polypeptide chain and you are mixing those chains. Suppose you have several threads and you are mixing them with each other. So you will have a complex kind of what we call that if we have like so many threads and they are interlinked to each other. We are not able to resolve it. What we call that? In Urdu, I guess we call it what Gucha, I guess. Right.
Sir.
So what are quaternary structure? Multi-subunit complex where each subunit is a distinct polypeptide chain. We have several polypeptide chain and they are mixed with each other, right?
Sir.
They will look like this. Here you can see this green one is a different polypeptide chain. This yellowish, bluish one is different, and this violet one is different. And these different polypeptide chains are mixed together. If you want to understand it even more better, suppose you had this secondary structure, alpha helix, you had this beta helix, you had this tertiary. You mixed all of them, right? You mix all of them, you will get quaternary. Or you will simply mix this type of, you are just mixing polypeptide chains. Now, are these four type of structure of protein clear? I would request you to just give it a read. Meanwhile, I will try to bring a detailed structure of protein where you can see the atoms. Try to give it a read. You have notes? You have the notes of this chapter or not?
Sir, school notes.
No, no, my notes.
Sir, let me check. I have, sir. I'll open.
See, these are different type of amino acids. This one is called tyrosine, this one is glycine, right? Different type of amino acids have different groups. The common part is they must be having a carbonyl group, the carboxylic acid group, plus amine group at alpha position. Apart from it, they can have different type of compounds as a side group. Suppose in this one, in this amino acid, TYR amino acid, we have a side group which is phenol, right? In this LEU amino acid, we have like tertiary this methyl group. So with different amino acids, we have different structure, and if we connect them, we will have different type of atom, and they will lead to different type of interactions or attractive forces, right? So when we are talking about sulfide bonds, we are using a amino acid which has sulfur atom. And I checked your homework. I asked you like what are reducing sugars. So what are reducing sugars? Your answer was very wrong. From where do you revise? I think you are not referring my notes. From where you are revising this chapter? Is my voice audible?
Yes, sir.
Okay. So from where are you revising this chapter? Are you referring some other notes? Because I have no problem. You can refer other notes as well, but the answer should be correct. The answer that you wrote in the assignment was wrong. The reducing sugars are those basically which can give positive tests for taurine and that taurine and silver mirror test, and Monosaccharides like free OH group, those kind of thing you were supposed to write. Okay. Is there a problem with these notes? So I can arrange some better ones.
Yes, sir.
So what is the problem? You can be frank. I will arrange better notes. If there is some problem, we can do better. Tell me the problem.
Yes,
Okay. So what are reducing sugars? Tell me you have the access of these notes. Try to read it because that question is very frequent in board exam. So you should know what are reducing sugars and what are non-reducing sugars. Are you reading the notes? Do you have notes or not?
yes, sir.
Okay, so what are reducing, non-reducing sugars? You can just read it from the notes. What's the problem, Ali? We need to move ahead. Try to tell the answer fast.
Yes, sir. I'm opening.
Okay.
Sir, all those carbohydrates which reduces Petri solution and tolerance reagents are referred as reducing sugars.
Okay, what else?
Sir, all
Which?
monosaccharides,
Mhm.
all monosaccharides, aldose and ketose are reducing sugars.
Okay, and if we are talking about disaccharides, what is disaccharide first of all?
So
You need to revise.
disaccharides,
Hmm.
they give 2 units of monosaccharides.
Okay, good. So, which type of disaccharides can be referred as reducing sugar? That
Uh,
is written.
if the if the reducing group of monosaccharides, aldehyde or ketone group are bonded, these are non-reducing sugars. So in
If
disaccharides,
Mhm,
if the reducing group of monosaccharides are bonded, then it is
mhm.
a non-reducing sugar.
Okay, so just
Sir,
let me tell you what was happening there. See, this is our aldehyde group, right? If this aldehyde group is free in disaccharide, then we will call it reducing. If this carbonyl group is not free, then we will call it non-reducing. Okay. And just because that one is important and the answer that you gave was so wrong, I am just going back. Okay, hmm. See, this is how sucrose looks like. This was the hydrogen which was anomeric hydrogen, OH. So this part is bonded, this part is not free, that's why it is non-reducing. I will show you how we are forming this sucrose after connecting, how we are basically connecting two monosaccharide units. I will teach you at the last of the chapter. So you don't have any type of confusion. So for now, let us move ahead. So how many types of structures do we have for protein? Four types: primary, secondary, tertiary, and quaternary. For primary, what we are talking about? We are basically talking about the sequence of amino acids in how we can arrange the amino acids, right? We have 20 type of amino acids. If we are arranging in a specific order, we are getting a specific protein with a specific biological activity, right? If we are changing the sequence of amino acids, so we are going to get a new protein. What are secondary? What are secondary structure of protein? If you are making this primary structure a bit complicated by bending it or by making a coil of it, you are going to get secondary structure. We have two types of secondary structure, alpha helix and beta plated. What is alpha helix? If you have this alpha kind of structure, then you will call it alpha helix. And if you have these type of sheets which are connected to each other by hydrogen bonding and they looks like what, folded sheets, then you will call them beta sheets. And if you are making this secondary structure even more complicated by even rotating it or bending it more, then you will get tertiary. And if you are if you are merging these all type of polypeptide chains, alpha, secondary, tertiary, or tertiary, if you are having a more than one polypeptide chain of these tertiary or secondary, then you will get quaternary. You can refer this example. You have different polypeptide chain and you are combining them with each other, then you are getting quaternary. Is this clear?
Sir.
So now we will talk about denaturation of protein. A protein found in living system with definite configuration and biological activity is called native protein. So what is a native protein? Which is found in our living system with definite configuration, biological activity. Okay. Now, what is denaturation of protein? The process that bring about changes in physical as well as biological property of the protein is called denaturation of protein. If somehow you are able to change the physical as well as biological property of a protein, then this process will be called denaturation of protein. Right? Now,
Sir.
important features of denaturation of proteins. Denaturation does not change the primary structure but changes the secondary and tertiary structure of protein. The sequence, the sequence in which the amino acids are connected after denaturation, you cannot change the sequence, but you can change the way how those chains are looking, like basically secondary or tertiary. Suppose you have a thread, you are making it alpha type, alpha kind of, if you are giving it alpha kind of structure, then you will call it secondary alpha helix. By denaturation, the secondary structure is going to change. The, the alpha helix will no longer look like alpha helix. How it will look like? Let me show you first. Suppose this portion, this portion is suppose alpha helix. After denaturation, how will it look like? This. Its shape will change, but the sequence in which amino acids are connected, it will remain same. Is it clear what I just said?
Sir.
Okay. You need to explain after this. So try to listen carefully. And if you have any doubt regarding anything, ask me again. Denaturation of protein can be caused by change in pH, temperature, or by adding other denaturation agent. If you are changing the pH or changing the temperature or adding any denaturation agents, you can bring denaturation of protein. Is it clear how you are going to cause the denaturation?
Sir.
By changing pH, temperature, or adding a denaturation agent. So, an example of denaturation of protein is boiling of egg. See, while you boil egg, this becomes into this yellow part albumin, right? So we will talk about it, how this is happening. So let us move it first. This is normal protein. This
Yes,
has
sir.
different type of structure. If we look closer to this, it is having alpha kind of structure, right? Alpha helix. If I see this specifically, it has sort of beta kind of structure, right?
Yes, sir.
But while I am doing what? Denaturation, it is losing its biological activity and converting into this denaturation protein. What happened to this denatured protein? Its secondary and tertiary structures are changed. The specific shape, the specific alpha helix shape that it used to have is no longer here. It has a different secondary or tertiary structure. Okay,
Yes, sir.
like here we have this. After denaturation, it loses its specific alpha helix secondary structure form and convert into this. So if the loss of biological activity is called denaturation, the regain of activity will be called renaturation. Is it clear?
Yes, sir.
If you are somehow able to convert this denatured protein into normal protein again, you will call that process renaturation. Denatured protein loses its biological activity. We have already talked about. Denaturation leads to increase in entropy. That means increase in randomness. This is what our albumin look like. So that example, let us understand it. Example of irreversible denaturation of protein is coagulation. In coagulation, what is happening? The albumin present in white part of egg. See, while we have normal egg, it has like liquid kind of structure, like liquid kind of thing inside it. While we are boiling it, while we are increasing the temperature, what is happening with the egg? It is getting denatured. It is converting into what? It is converting into this solid form, right? This is what?
Yes, sir.
Coagulation, and it is a part of denaturation of protein. Curding of milk is again denaturation of protein. What is happening while we are forming curd? Uh, which is caused due to the formation of lactic acid, bacteria, basically bacteria and stuff, right? Lactic acid. In milk, we create lactic acid, and due to which bacteria and other stuff lead to the formation of curd. Uh, Basically, denaturation process. Is it clear?
Yes, sir.
So, in simple terms, can you tell me what is denaturation of protein in simplest term, in 2 or 3 sentences?
The denature of protein, it affects the secondary, tertiary, and quaternary, but it
Mhm.
doesn't affect the primary.
Good, good. What
So
else?
it doesn't affect the proteins. The uh, and their denaturation proteins.
Mm, see, try to say this now. How can you cause the denaturation? By changing the pH, temperature, or
Yes,
adding denaturation
adding
agent,
agents. Yes.
right? Denatured protein loses its biological activity. See, if you are causing the denaturation, what will happen? They will lose their physical
Biological
as well as
activity.
biological property.
Yes.
So if you write these points, then you will get sufficient marks. And you can add this as well. It leads to increase in entropy. Why increase in entropy? Because while you are increasing the temperature, the particles or the molecules inside that protein will have thermal energy. If they have thermal energy, they will try to move, they will try to vibrate, and due to which entropy will increase. Entropy is what? Randomness. Okay.
Sir.
Is it clear what is denaturation of protein?
Yes, sir.
Now, protein is complete. We have nucleic acid. Just give me 2 minutes, then we will talk about nucleic acid. Meanwhile, revise what we have studied so far. Just give it a read. See, this is albumin in our egg, this yellowish part in liquid form, right? While we are heating it, what is happening? While we are heating it, it is converted into egg white. That solidish opaque type of structure, right? So
Sir.
let me just copy paste what is happening. Give me a second. Okay, just because I change this form here. I will add a clear, like, the thing is same. The egg, this albumin will be converted into egg white, and it will lose its biological property, and the primary structure will be same. The secondary and tertiary structure will convert. I will write it somewhere. Okay, here it is not in that much of detail. I will write so you can understand better. You get the gist of it now. What is happening while we boil egg?
Yes, sir.
The albumin, this protein convert into egg white, which is consumable. So now let us talk about nucleic acid. All right. The particle in nucleus of the cell responsible for the heredity are called chromosomes. What are chromosomes? Which are responsible for the heredity. What is heredity? The, like, how the traits are transferred from the father to son, right? Which
Yes,
are
sir.
made up of proteins and another type of biomolecule called nucleic acid. So, what is nucleic acid? The particle in the nucleus of the cell responsible for the heredity are called chromosomes, which are made up of a protein and another type of biomolecule called nucleic acid. Chromosomes are made up of nucleic acids, right? And chromosomes are what? Chromosomes are present at the nucleus of cell, and chromosomes are responsible for the heredity. And chromosomes are made up of protein, which is nucleic acid, right? What
sir.
is nucleic acid? A protein present inside chromosome. Chemical composition of nucleic acid. Nucleic acids are basically of two types, DNA and RNA. What is DNA? Deoxyribonucleic acid. And what is RNA? Ribonucleic acid. And DNA and RNA are two type of nucleic acids. Let us talk about it. The monomeric unit of nucleic acids are called nucleotides. What do we mean by monomeric unit? Nucleic acids are formed by combination or by connection between nucleotides. The smallest unit inside the nucleic acid is what? Nucleotide. In the similar way, while we were talking about carbohydrate, what was the smallest unit in carbohydrate? Monosaccharide. After connecting monosaccharide units, we were getting different type of carbohydrates. Here, after connecting different type of nucleotides, we will get nucleic acid, right? What is
Yes,
the
sir.
monomeric unit of nucleic acid? Nucleotides. And where are nucleic acid present? Nucleic acids are present in chromosomes. Okay.
Sir.
Each nucleotide consists of three parts: a phosphate group, a 5-carbon sugar, and a heterocyclic base. A nucleotide, which is a monomeric unit of our nucleic acid, consists of three groups: phosphate group, a 5-carbon sugar, and heterocyclic base. These nucleotides, after connecting with each other, will lead to the formation of nucleic acid. This single unit of nucleotide will connect with different nucleotide and they will form a nucleic acid. So let us understand what is happening. Nucleic acids are also called polynucleotide. What do we mean by polynucleotides? Where several units of nucleotides are connected with each other. Is it clear?
Sir.
One more thing. While we will be uh, drawing the structure of these nucleotides, we will always see the sugar is at the middle. So we can say sugar is always in the middle of our phosphate group and heterocyclic base. It will look like a sandwich. At the top, phosphate group. At the center, we will have our sugar. Then our heterocyclic base. We will see how this structures look like. Nucleic acid, either it is going to be DNA or RNA. The smallest unit of nucleic acid is nucleotide, which consists what? A pentose sugar, a phosphoric acid, and a heterocyclic compound, which is base. Complete hydrolysis of DNA or RNA yield to pentose sugar, phosphoric acid, and nitrogen containing heterocyclic compound called base. If you are hydrolyzing, if you are doing the hydrolysis of nucleic acid, you will get the units of nucleotides. This is basically what? This pentose sugar, this phosphoric acid, and this nitrogen containing heterocyclic compound bases. What is this? These are our nucleotides or not? Nucleotide is
Sir.
basically these things. So if nucleic acid is composed of nucleotides, if I break nucleic acid, what I will get?
Nucleotides.
Nucleotides. On what nucleotides consist? These three entities. Is it clear?
Sir.
Just give it a read. And if you have any questions, ask me. And please revise from my notes because here everything is written in very concise way. Out of it, you won't see any question. If there is any part you are not able to understand, then ask me.
That's all.
Done? Any questions?
So,
Okay. In DNA molecule, the sugar moiety is beta D 2-deoxyribose. This sugar moiety. In every nucleotide, we have three components, three entities. First one is phosphate group, then our sugar, then heterocyclic base. The sugary, this sugar part in our DNA is this thing. This sugar part in our DNA is this. Let me write it clearly. CH2. Okay,
so
this is looking like Harvard structure that we studied in glucose while we were forming the ring of glucose. We were getting this kind of structure. To be more specific, this looks like fructose structure because we have 5 member ring, right? Or not?
so
Now, the sugar part of nucleotide in our DNA look like this, and this is called what? Beta D2 deoxyribose. The sugary part in our nucleic acid, to be more specific, in nucleotide for DNA molecule look like this, and this is called beta D2 deoxyribose. In your exam, they will give you several options. They might give you several structures, and they will ask you, out of these, which sugar molecule is for DNA molecule. So you will have to identify this one is for DNA. And if they ask you about RNA, you have to pick this one. For RNA, the sugar moiety is called beta D ribose. For DNA, the sugar entity is called beta D2 deoxyribose. So by the name, we are going to understand what is happening. Here we are saying deoxy. If I am saying deoxy, some of the oxygen group is going to leave their respective site, deoxy, right? So here at the second carbon, if you relate it with this one, in the second carbon, we have OH group here. That's why we are calling it what? D-ribose. Here at the second carbon, this OH group is not present. Here we only have H. So that's why we are calling it deoxyribose.
so
So after this, I am going to connect all of them. I will connect the sugar part, the phosphate part, and that base part, and I will connect it with a different nucleotide. So try to draw it once. Draw both of them in your notebook because they might ask you which out of these structure is for DNA and which is for RNA. This is the sugar part.
so
Done?
so
So why we are specifically using this 2? This 2 is to show that the second carbon is losing the oxygen, deoxyribose. Is it clear? The
Sir,
DNA, beta D2 deoxyribose, for RNA, beta D ribose. Now, DNA contain 4 bases. Our DNA have 4 bases. You need to learn this. I used to learn this like AGCT and AGCU. Adenine, guanine, cytosine, thymine. AGCT for DNA, for RNA, the first 3 are same and the fourth one is uracil. AGCT and AGCU. DNA contain 4 bases which are adenine, guanine, cysteine and thymine. This again is very important. They will directly ask you, tell which kind of bases are present in DNA. So you need to write adenine, guanine, cytosine, thymine. If they ask you what type of bases are present for RNA, you will say adenine, guanine, cytosine. For last, you will change thymine with uracil, AGCU. RNA
sir.
also contain 4 bases. The first 3 are same as in DNA, but the fourth one is uracil. Okay.
Sir.
What we talked about here? We talked about the sugar moiety of DNA, the sugar moiety of RNA. Why we are talking about B? Because these are the basic component of the entire nucleic acid molecule, right?
Yes,
Because
sir.
the smallest unit of nucleic acid is nucleotide and nucleotide consists these things. So we are talking about them as well. Now, structure of nucleic acid, primary structure of DNA. We are going to talk about how the structure of DNA look like. Now we are going to connect this sugar phosphate base and heterocyclic part with each other. Let us understand what is happening. The structure of nucleic acid depends on the sequence of nucleotides. Depends on sequence of nucleotides. What are nucleotides? Nucleotides are the smallest unit of nucleic acid. The the connecting link in nucleic acid is phosphodiester linkage. Again, in our carbohydrate, the linkage was glycosidic linkage. In our protein, the linkage was peptide linkage. In nucleic acid, the linkage is which type of linkage? Phosphodiester linkage. Okay. In carbohydrate, the linkage was which one? This is again important questions. They will ask you the type of linkage in 1 mark. In carbohydrate, which type of linkage we are using for carbohydrate? For carbohydrate, which type of linkage we use?
Sir, sir,
Glycosidic linkage.
glycosidic. Yes, sir.
For protein,
For protein, pep- no, peptide linkage.
peptide linkage. For nucleic acid,
Phosphodiester.
you need to remember these three linkage kinds. Okay. So the connecting link in nucleic acid is phosphodiester linkage. The information regarding the sequence of nucleotide in the chain of nucleic acid is called primary structure of nucleic acid. The information regarding the sequence of nucleotides, how the nucleotides are going to be arranged in the chain of nucleic acid is called primary structure of nucleic acid. In the similar way, while we were talking about proteins, in proteins, what we were observing, the specific arrangement of amino acids was responsible for the structure of which type of structure? Primary structure. If I am changing the sequence of those 20 types of amino acids, I am getting new protein, right? In the similar way, here, if I am changing the sequence of nucleotides, I am going to get different type of nucleic acid. For example, I have this nucleotide, right? And I have this nucleotide. I am connecting these two. The sequence is like this: black, yellow, and black, right? Again,
Sir.
give me a second. See, I have this black nucleotide, this yellow nucleotide, and again black. If I am going with this sequence, I'm going to get a specific type of nucleic acid. But if I'm changing the sequence, similar to what? Proteins, I am going to get a different
Yes.
type of nucleic acid. The sequence matter. And this smallest unit is what? This smallest unit, which is responsible for the formation of nucleic acid, is called
Nucleotide.
nucleotide. Okay.
Yes, sir.
A simplified version of nucleic acid chain is shown below. See, this base, sugar, and phosphate is what? These moieties connected with each other is what? Basically a nucleotide.
Yes, sir.
And if I am connecting several nucleotide units, what I am getting?
Nucleic acid.
If I am connecting several units of these nucleotides, I am getting nucleic acid. Now, the bondage or the linkage is always going through what? Phosphate and sugar. And this linkage is called phosphodiester linkage. Is it clear?
Sir.
Now, we will talk about nucleoside and nucleotide. Again, important. Everything which is written in these notes are important. Okay, so you need to learn everything. I think these are of like 10 or 20, 10 or 15 pages, so you can do this. So now we have this nucleotide. We knew nucleotide. What are nucleotides? We have understood so far. If we have a sugary part, a phosphate part, and a base, if I am connecting them with each other and I am putting the sugar in the mid part, like a sandwich, then I will call it nucleotide. But now we have this new thing, which is nucleoside. So let us understand what is nucleoside and how it is different from nucleotide. So a nucleoside is what? A unit formed by the attachment of base to one position of sugar. In nucleoside, we only have two moieties. In nucleotide, what we were having? We were having our phosphate group, our sugary part, and a base. Let me see. Just give me a second. See, try to understand it. Uh, nucleoside is a monomer of nucleic acid. We had what? We had our phosphate plus sugar plus base. Okay.
Sir.
These three moieties connect with each other, putting the sugar in between and form nucleotides. If if I am removing this phosphate, if I am removing the phosphate moiety from this nucleotide, what I am going to get? A nucleoside. If I am removing this phosphate part, if I am suppose here, if I am if I am removing this phosphate part and I only have these two things, then it is what? Nucleoside.
Right. Yes, sir.
Okay. Now, so what is nucleotide? When nucleoside, what is nucleoside? Nucleoside is sugar plus base. When nucleoside is linked with phosphoric acid at fifth position of sugar moiety, it is nucleotide.
Nucleotide. Yes.
Nucleoside plus phosphoric acid, we will call it nucleotide. Okay, this is very easy and they have asked this in the board exam. So try to remember it. Now let us uh, like connect all the things we have studied so far. Like how does the phosphate part look like? How does the sugary part look like and how a base look like? See, here, this is nucleoside. This is our base. This is our base, which is connected
Yes,
here.
sir.
And just above, we have talked about how the sugary part of our nucleic acid look like. This is what? This is how the sugar moiety look like, right? In
Yes,
nucleic
sir.
acid, this is how the sugar moiety look like. If I am placing base at the first position here, then I will call it what? Nucleoside. Here we have what?
Nucleoside.
This sugar plus this base. It
Yes.
is nucleoside. Now, if on in this nucleoside, if I'm able to add this phosphoric acid part as well, in this nucleoside, if I'm able to add this phosphoric acid part as well, then it will be converted into what? Nucleotide. Suppose at the carbon number 5, you are able to add this phosphoric acid part, then this is now our nucleotide. And if you connect these nucleotide unit with each other, then you will get what? Nucleic acid. If you have this, suppose if you have this, and you have connected another, if you have connected what? Another unit of the same thing with this bond, then you will get a nucleic acid. Let me draw it for you, right? See, here, they were connected with this part, right? This phosphate part will be connected with the new sugar part of this, a new nucleotide. Is
Sir.
it clear? Do you have any doubt?
Yes, sir. No, sir.
This chapter is almost over. Please try to read it for 2 minutes, then we will move ahead. If you have any doubts, please ask me. And you will be having a test from this chapter. So prepare and try to read it from my notes because here it is written in very crispy.
Sir, it always has to be linked at 5-degree position. No,
Yes,
like fifth position
yes.
every time.
This phosphate part should be at the fifth position because
Okay, sir.
try to understand it. This sugar part is going to be same always. This sugar part is always going to be same for DNA. For DNA, the sugar part will look like this.
Sir.
Always for
Yes,
RNA,
sir.
this sugar part will always look like this. So obviously at the fifth carbon, will be having our phosphoric part always.
Sir.
Okay. Any other question?
Sir.
Or
Yes, sir.
okay. So let us move ahead. Now, double strand helix structure of DNA. This is just a representation of DNA in a better way, in a way where we can understand the properties of DNA even better. Okay, so let us understand what is this double strand helix structure of DNA. James Watson and Francis Crick gave a double strand helix structure for DNA. You two nucleic acid chain are wound about each other and held together by hydrogen bonds between parts of bases. The two strands are complementary to each other because the hydrogen bond is formed between specific pairs of bases. Adenine forms hydrogen bonding with thymine, whereas cysteine forms hydrogen bonding with guanine. In secondary structure of RNA, helix present, which are let us understand directly with the structure. See, this is how our doubly helix strand structure of DNA look like. What is happening? What we have here? Two nucleic acid chains, and they are
Yes, sir.
wound about each other and held together by hydrogen bond. See, I have this nucleic acid chain, and I have this another nucleic acid chain. They are wound up upon each other, and the bonding that is present inside them is which type? Let me mark it. The hydrogen bond is formed between specific pairs of bases. Adenine forms hydrogen bonding with thymine. Cysteine forms hydrogen bonding with guanine. The thing that I have underlined is a question which is asked in boards. They might ask you, the question is the two strands are complementary to each other. Why? So you need to answer because the hydrogen bond is formed between specific pairs of bases. Adenine forms hydrogen bond with thymine, ATCG, like this. And in the structure, how it is going to look like. This is our DNA structure. And if I'm going to talk about the bonding, how the bases are connected, try to look over here. The blue part is cysteine. This blue one is cysteine. The green one is guanine. This yellow one is adenine, and this one is thymine. Try to focus on these parts. This is two different bases which are connected with each other. This red part is what? This red part is a base. This base is which one?
I mean,
And it is connected to
adenine.
adenine. And if we look here, this green one is guanine.
Guanine.
And this one is connected to
Cytosine.
which one?
Cytosine.
Cystosine, right? So what we are observing here, this thiamine is connected with adenine, and am I writing it right? Thiamine is connected with adenine, and cysteine is connected with this. So what we can say, ATCG, right? So what is happening while we are talking about double strand helix structure of DNA? Simply, we will have two chains of nucleic acid. They will be wound up around each other, and there will be hydrogen bonding between bases. Which type of bases? Adenine will form hydrogen bonding with what? Thymine, and our
Thymine.
cysteine will form hydrogen bonding with guanine.
Guanine.
This is all about our double strand helix structure of DNA. It was given by James Watson and Francis Crick. Try to give it a read. This is important. And if you have any question about this, you can ask. Any part, any term that you are not understanding, ask me. This is the question. The two strands are complementary to each other. Question mark. The answer is because the hydrogen bond is formed between specific pairs of bases. AC, ATCG. You can learn it like this.
Yes, sir.
Is it clear? Any question?
Yes. No, sir.
Sure.
Yes, sir.
Now, in a similar way, let us talk about the structure of RNA. So how does RNA will look like? RNA will look like this. In RNA, what is changed? We only have one chain of nucleic acid, right? Due to which we will only have bases which are not connected with other bases because we only have one chain. Here we have this base because we don't have any other chain, so it is not connected with anything, right? So this is how our RNA structure look like in double strand. So you can write it like this. The secondary structure of RNA helices are present which are only single-stranded. What do you mean by single-stranded? They only have one chain of nucleic acid. Sometimes they fold back on themselves to form double helix structure. Sometimes what will happen? They will fold back on themselves. Suppose you have this chain, they will be folded and they will form bond with each other like this. Here, they will fold like this, and due to which these two bases will be near to each other and they will form bonds, right? So you don't have to go there because in your NCRT they haven't shown any structure regarding that. So if you draw like this, it is sufficient. And to be more specific, they will only ask you about the double strand structure of DNA. They won't ask you about RNA, right? So you need to
Yes,
be
sir.
very clear with this one. You need to be very clear with this one, right?
Yes, sir.
Now, just miscellaneous part, types of RNA, messenger RNA. This was a question that was asked in my exam while I gave my boards now. So important again. Types of RNA, messenger RNA, ribosomal RNA, and transfer RNA. Right? Simple. Biological
Sir,
function of nucleic acid. Yes,
sir, so in the question it asks what is mRNA? Like, how did the question ask?
they will ask you types of RNA. So you will say messenger RNA. So messenger RNA is what? It is basically transferring the data. So let me do one thing. I will add like like few detail about these as well. What is messenger RNA? Or let me write it. Just give me a second. Let me copy paste it. Give me a second. I'm adding the details about it. Just give me a second. Okay? Mhm. Is my screen visible?
Yes.
So messenger RNA is simple, like is a type of RNA that carries the genetic instruction from DNA to ribosomes where proteins are made. Simple, you can write it. Right. What are they? They are basically messenger. What they carry is genetic instruction from DNA to ribosomes, right? What is ribosomal RNA? Is a type of RNA that forms the core structure of ribosomes, the molecular machine in cell that synthesizes protein, right? What is
Sir.
transfer RNA? As the name suggests, is a small RNA molecule that plays a critical role in protein synthesis by carrying amino acids to the ribosome and matching them to correct codons on the RNA. See, if you write like even this, you will get 4 marks. Otherwise, like if you write the types now, they will ask you what are nucleic acids. So you will have to write RNA, DNA. In RNA, you will write how RNA sugar moiety looks like, right? How the nucleic acids are formed by nucleotides. What is nucleotide? Nucleotides have phosphoric link, phosphorus part, sugary part, and that base part. And to like get even more marks, you can write the types of RNA as well. You won't be needing this. Otherwise, like you can read it. It's simple. By giving it one read, I hope you can understand it.
Yes.
Now, last biological function of nucleic acid. Nucleic acids are prefer. Nucleic acid performs two important functions: replication and protein synthesis. That's it. What is replication? It is the property of biomolecule to synthesize exact copy of itself, right? And
Sir.
that's it. What is protein synthesis? Protein synthesis is the synthesis of protein. Is this clear?
Sir.
This was about our biomolecules. We have like two very small topics, vitamins and enzymes. It will take like 15 to 20 minutes. So are you free? Do you have any other classes after this?
Sir.
You have other classes?
Sir, I don't have a class.
Okay, just give me 2 minutes and let us complete this. Hmm. Or we can do one thing. We will do the enzymes and vitamins in the next class. It will take like 15 to 20 minutes and we will start the next chapter as well.
Sir.
So prepare till here. You will have a test of this chapter till nucleic acid, right?
Sir.
And for now, I will share this updated PDF as well. If you
Sorry.
have any doubts, you can ask now.
No, sir.
Sure. And just a
Sir.
question, like in the assignment, you answered the like that. What was the question? Reducing sugars. From where did you answer that?
Sir, like in the
What
notebook
was the
and
source? What book?
some notebook school.
Try to avoid that answer. That was not correct. Okay. You have to answer from this notes if you need.